back then the enzyme was extracted from corn. It was nitrate reductase, and the goal was to be able to come up with a platform where you could produce and purify the enzyme, and purify the enzyme in one step only to decrease the cost. So that's kind of how I got into the meat of protein expression purification in the industry and cost, because that's all they care about is cost. So that's the main challenge, is cost, always. Then from there, back and forth, I worked into metabolic engineering in the French companies, and I went back to the US, and I worked at Novozymes for a while. Novozymes is a big industrial enzyme company. It's one of the leaders in the market. It's making mainly commodity enzyme. I'll get back a little bit to it. And I currently work for JoynBio. It's a little bit different. I'm working on engineering microbe for agriculture. Still some protein work involved, a lot of it, since it relies on nitrogenase. And I'm working in the same building, in the same lab as this company, which is also a protein production company. So here's a quick rundown of my background. So main industrial challenges, like I said, it depends. Everything is about money when you get in the industry. If you want to have a protein and sell it, if it costs $1 million, nobody's gonna buy it, unless obviously you have different market. So in one case, in the case of, in the case of pharmaceutical company, you have a market which is pretty big, it's worldwide, and a lot of money. So that's a challenge, and that helps also for everything else, all the scientific challenges we come across, because the cost is not going to be an issue in that case. So in that case, you're a specialty compound, like we call it. On the bioprocessing, it doesn't really matter if it's intracellular or secreted, because the cost of the bioprocess and the production has a limited impact on the overall cost of the enzyme. It's such a high value that it does not matter what you do at that point. So you need to work your economics right before making this kind of choices, but often that's the case. I'm gonna talk more about what I know, which is commodity enzyme. So the market is fairly high. Usually it's still a worldwide market. It's not low value. The application is commodity. I'm talking about price ranging from $2 to $3 per kilogram of purified enzyme, right? So if you're in the pharmaceutical business, a kilogram of protein, I don't know how much that the retail cost is for, but probably millions or even more. So that's gonna put some constraints on your bioprocessing. What I call bioprocessing is obviously all the process to generate the product. In that case, you're gonna look at secreted proteins, most likely recombinant. You have barely wild type, you don't find many wild type protein from the wild type host. You still have some exception here and there, but it's recombinant. And we are talking about that type. This is actually what you produce here. And this is where you find it. So again, your enzyme has to be cheap, because if you want to put one of these little doses of detergent in your dishwasher, if it costs $50,000, might as well buy new stuff. So in that case, the cost of production has a major impact, right? So I'll talk a little bit later about that, but right now we'll focus a little bit more on the protein. So you want simple folding, so most likely no disulfur bonding, because it's hard to get that going. You need something really stable. So you can imagine from production to there at the application of the bioprocess, it needs to be stable. So monomers or homodimers or polymers of same subunits are preferred. You don't want any cofactor like cytochromes and things like that, because first it's expensive and you need a co-filling for it, and second, well, it's really hard on the folding on the extracellular space to get that put together. And as much as possible, only in some cases, no post-translational processing. You can have a pre-enzyme which gets activated, but you don't want glycosylation or things like that. So this is actually a real factory, enzyme production factory. So that shows you a little bit of the challenge. You get from here to there. So what are the main enzymes? So you have what we call, so these are commodity enzymes, technical enzymes. So you find them in detergents, you find them also in food, animal feed, like Dick was talking about, diagnostic enzymes, so glucose oxidase for the diabetic sensor. You have biocatalysis enzymes, so these are process enzymes found in chemical productions, like invertase to make high fructose corn syrup and things like that. Or you have what we call bioraffinery enzymes, so mainly amylase, endoglucanase, and cellulase. They spread through a wide class, a lot of classes, so I'm not going to go into it. I just want to pinpoint that this is a huge market. This is what you find in detergents, so those dish soap or laundry detergents. You have protease in them to get rid of the stains. And this is a pretty big market. A lot of companies are working on that. This is also more bioraffinery and food. I'm not going to go into it, but if we stay on the detergent market, you can imagine how lipase to get rid of fat stains is really important on the dishes. So the list goes on, and I'm not going to go through all of it. On your application, you may have different operating conditions. So, for example, if you're in the feed, so here you have the temperature, here you have the pH. So in the feed, you're gonna be slightly acidic because it's intestine and temperature around 30 degrees Celsius. But for the starch, you might be operating at temperature around 90 degrees Celsius baking. Well, when you bake stuff, obviously you put it in a oven. Same thing for textile. And then on the other hand, you can have the food industry, which usually operates at low temperature to prevent contaminations, but you still need to have enzyme active around 10 degrees Celsius. So you have a wide spread of application and conditions. So this also is a huge challenge to find an enzyme which is gonna work for your particular application. So now let's say you have one enzyme. You know you have one protease, one endoglucanase, but you have different applications requiring different conditions. So the first thing you can do before getting into engineering your enzyme is to look in the physiological diversity. So for example, this is an example of an enzyme, I can't remember which one, but through the physiological diversity, you have different range. So that's already a start. You know your enzyme, you know all the restrictions around your enzyme, what you want for being a good secretion, being secreted correctly. And you have different applications. So first you look into the physiological diversity. And you'll pick the one which already as a wild type seems to be working for the best conditions, for the conditions of your particular application. Where do you get that? Well, nature is a tremendous source of enzyme diversity, okay? And most of them, we find them in microbes. So microbes meaning from all eukarya, bacteria, archaea. So they break down and recycle organic material in nature, so you know you're gonna have a good source of enzyme. They are secreted, which makes it fairly easy to test. Some of these bacteria already naturally have high ability to produce the enzyme in high yield, and it's easy to collect, store, grow in the lab. So, for example, this is for breakdown of cellulose. You can look into the bacteria which degrade the leaf, on this mushroom which are growing on the side of the trees, or in the textile eating and bacteria. So if you fold something and you haven't touched it for 50, 30 years and suddenly you open it and you have big holes, somebody digested your cellulose. So that's also a good starting point. Novozymes. So most of the activities I'm talking were also linked to novozymes. is doing bioprospecting, so going all over the world looking for microbes in different conditions. From there, you get a culture collection. So you have more than 10,000 to 50,000 microbes in the collection. So that's kind of old school because it was still physiologically based where you have a culture collection. And from there, you're gonna take all these microbes and put them with the substrate you want and induce the expression of a given enzyme by inducing, for example, in the case of cellulase, you put some cellulose as a carbon source, and you try to see which one are growing or not. Then if you get a fungal mycelium in that case, because most likely in the case of cellulase, it's a fungi because they secrete it, saprophyte, and they're gonna eat up leaf material, wood, and things like that as carbon source. You extract the cDNA, so you don't know which one, right? Because there's a bunch of cDNAs. You put it in an expression vector. Sometimes you put it in yeast or other system, and you do a high throughput screening to find the right enzyme and link the gene to the enzyme. So this is how a lab looks where I work currently. You can see that's about a third of the lab, and you have 4 of them like that. So it's super high screening and robot based. One of the main issue when you do enzyme screening so cellulose is fairly easy, but let's say protease is to find the right substrate. So you have chemical synthesis group which are there to find the best substrates which are going to be predictable to your application, because an enzyme activity is nothing if you use the wrong substrate. So even if you have a good enzyme which works really well on your substrate, but this substrate is really far from the actual natural substrates you want your enzyme to work on, your whole screening is worthless, useless. So now, you have an enzyme, you have a gene, you know what you want, and you need to go from the milligram to that level, and you want it cheap. So milligram per liter, at the industrial scale, you usually get up to 50 to 100 grams per liter of protein, and I'm talking about 50,000 liter fermenters. And that's about what you need to get a low cost for your enzyme to be found in applications. So how do you improve the expression? Well, Dick went through a fairly thorough list of what you can do, but one first, it's you check for the diversity in nature. Sometimes you're gonna find stuff, enzyme, which gonna express and secrete better than other, just naturally. If you narrow it down to one candidate enzyme, you can improve the specific activity. So you can do protein engineering with 3D structure. I'll be honest with you, it never works. It's useful to understand what's happening, but people have been using random mutagenesis because you can use robots and you'll see what works. Because of the high throughput and the screening, it's actually faster and cheaper nowadays to do random mutagenesis and protein engineering. It depends, right? It depends exactly, because sometimes you identify some mutations which are true for a whole class of enzyme, and so you can just right away do that and have a targeted mutagenesis. And you can have both. So on the host side, which one's gonna work the best? Well, it depends where your enzyme, the source of the enzyme, the origin is coming from. But mainly it's trial and error. And you also have patents sometimes which can block you and prevent you to use one host for one specific enzyme because a competitor already has done it. So you cannot, even if it's the best way. And then here it's kind of the same thing. You do genetic engineering of the host to increase the total activity by improving, for example, working on the secretion pathway. Or you can work on removing the proteases. And one thing when you work with hosts which are secretion hosts, you want to remove the background activities to make sure that your product, your broth, is going to be as pure as possible. So you're going to knock out the proteases and all the native enzymes which are naturally secreted by the enzyme if it does not affect its growth. And you can also improve the expression by random mutagenesis. So let's say you have a good gene, you have a good host, and you're trying to get to the next step. You have 20 grams per liter, you want to move it to 30 grams per liter. Nothing rational has worked. You're gonna go through a random mutagenesis campaign. So there are strains like that at Novozymes which have been going through this process for 30 years. And each time they have milestone strains and they can improve it. So this was a little bit, things have been changing with synthetic biology. Okay, so nowadays you can just order the gene. People are making the comparison between biology and electronic engineering. A lot of money has been poured into that, and that's what Ginkgo and the company I'm currently working with are mainly using for improvement. So the way it works, it's a platform where you design. So you have a whole up front. You do a design of your genes, and there you can try different promoter, different ribosome binding site, different codon optimization. You have no limit. So if you have 1 or 2 enzymes and you want to try different combination, you can easily generate 500-600 constraints. So it's great, but if you cannot synthesize that, how do you do it? We are lucky enough to have on-site in the company a DNA synthesis lab, and we also work with other companies. So from design to build, it's about a month to 2 months. And it doesn't matter if you have 1 gene or 10,000. So often we go and we order 10,000 genes at once. So then you have them. Then after you need to test, and that's where the robots are useful, because you have automatized transformation and all the liquid handling. So each time you take some liquid from one plate to another one, it's done by robots. And you can automatize the transformation, the screening, the storing. So it's pretty cool. It works really well. Still some issues, but overall it works. And then after you test, and you can ferment. And same thing for fermentation. It used to be a bottleneck, but nowadays you can have this fermentation around which have up to 24 fermenters you can run at once. So I'll talk about that a little bit later today. So yeah, you have custom software, automation, and next generation sequencing is really the key here. And that's how the lab looks. It's kind of a little bit disappointing when you get there, because it's just stack of machine instruments stacked on top of each other and almost nobody working in the lab. So I know that we have another session by William on enzyme degrading plant cell walls, so I figured out that would be a good way just to show an example of what's been done. So Novozymes is a leader in to manufacture cellulase for the second generation bioethanol. So where you take directly the biomass and turn that into fermentable sugar. So either glucose or xylose, C6 or C5 type of sugar. So already right there you have a lot of challenges, because you're gonna be working. So you take the biomass just quickly. That's corn in the US mainly. So it's not sugar cane like in Brazil, it's corn here. Then you treat it, and you have different pre-treatment. It's still chemically, harsh treatment, high temperature chemicals. And then you have an enzymatic hydrolysis where you get to this little powder, which is a fermentable sugar. So you'll see this thing, sometimes I use it as a sugar and sometimes I say it's enzyme. In that case, it's sugar. And then you ferment and you're gonna make ethanol from there. Excuse me. So here you have a lot of challenges, because you have something which contains a lot of inhibitors. Sometimes it's not really well rinsed, so you have a lot of harsh chemicals as well. So you need to find the right enzyme for that. So quickly, cellulase, you have more than one enzyme. You have at least 3 enzymes. So you have the 4 enzymes. So you have the cellobiose hydrolyse. So you have 1 and 2, depending on if they work on the crystalline or the amorphous zone. And you have endoglucanase, also as well as beta-glucosidase. And you also have a bunch of other proteins which are helping all this. So either for redox or sometimes for the physical removal of the enzyme from the cellulose. So it's a blend of many enzymes. So that's the kind of diversity you get, so you can imagine all the enzyme you need to get to one unit of that, or one of this. And that's how it looks in, I mean, that's not the real stuff, but that's kind of how it looks. And you have lignin, which are polyphenols and are extremely inhibitory to either the yeast or to some of the enzyme degrading the cellulose. So you also need enzyme to go and cut that because it's cross-linking the cellulose to make sure it stays as a good fiber, really strong. So other constraints, not only scientific, but based on the production or the application. So here, this is the enzyme storage at a factory, okay? So it's outside, so it has to be stable. There's no way you can cool that off or heat it. It would be way too expensive. So it has to be stable here, and it has to be usable, and it also needs to be safe, right? So it must be cheap, again, because the major cost of a 2G bioethanol plant is the enzyme. It's getting efficient, but it's still pretty expensive. So you need to keep the cost down in order to have people to adopt ethanol. So it has to be cheap. Lot-to-lot dosing must be the same. So you cannot have fermentation batch where you make a lot and suddenly you don't make much. So it has to always be identical. QA/QC is really important. And the enzyme also must be active in the process condition, low pH, medium temperature. So here the processing of the enzyme, not the processing, but if you look at the enzyme hydrolysis, you get things coming directly. It's not being cooled off, so it can have temperature of about 100 degrees Celsius, and you have some HCl, some added to help, and it stays there for an hour. So your enzyme has to work through all these conditions. So this enzyme, in the case of cellulase, are a lot of different enzymes. So you're going to have different expression hosts making different enzymes. And then after you take all this broth and you combine them together, you blend them, you mix them. So how do you optimize this? Well, obviously you have the blending optimization. If you have a well-known broth enzyme, you can change based on the enzyme, the components of this broth, you can change the ratio of one broth to the other one. But if you look at one expression host or one enzyme, you need to improve the expression. So the host is gonna be essential. So in that case, we use trichoderma, because this microorganism naturally secrete a lot of cellulase because it's a saprophyte which feeds plants, plant material. So you optimize it, so that's kind of the wild types host. But you can optimize the secretion, you knock out all the proteases, and you knock out all the secreted enzyme, including the native cellulase, because you don't want this native enzyme to mess around with your enzyme. You want something pure and clean. Then after the gene expression, well, again, you can modify the copy number, the promoter, the codon frequencies, ribosome binding sites. So that's the thing you can do for the expression. When it comes to the activity, again, same as what I was showing earlier, site saturation mutagenesis of selected target you already have defined through the random mutagenesis, or you can do a diversity screen and look in nature to get better enzyme. I'm just showing quickly, these are different, so these are the different enzymes and different substrates. The goal here is not to get into the result, but it's to show that when you look at the units per milligram of protein, you can see that through the different type of mutations or modification, you have different activities, right? And so that kind of illustrates the the optimization of an enzyme. So it's a lot of work put in one single graph. This takes about, it started by a cellulase blend which was originally used for textile. So they started from there, and then through the different years and iteration of optimization, got to that point where it became to be a commercial product. It was meeting all the requirements for the commercial product in terms of cost and stability. And that's an improvement of about 6 times. So you have to put 6 times less enzyme to get to the same result, pretty much. So it's a mix of protein engineering, host engineering, random mutagenesis. And so this allowed, so these are the progress done, so it can work on corn, on wheat, on sugarcane, on wood. So you have the improvement here. Anyway, long story short, it ends up being a cheaper cost of operation, which increases the profit of the manufacturer, and in the end, ultimately decrease the cost of the production of bioethanol. And the low cost allows to start competing or blending with petroleum. So we are talking 2G, right? So from cellulose, not from starch. And eventually can lead to energy independence. So in Brazil, I took this example. I actually went there to this particular plant. This is a starting material, and by adding what we call a bolt-on, so a module you just add to the pre-existing plant, so something cheap for the manufacturer, they were able to convert it to a 2G ethanol plant. And Novozymes worked a lot with this company in order to get there and make the price cheap and make it a commodity compound. So I'm going to switch gear a little bit to talk about enzyme purification and its constraints and the constraints of the bioprocess applied to the enzyme. So this is how it looks, a simple diagram. So you start from your fermenter. So we are talking about enzyme being secreted. So they are in the broth. So you have your host and your protein. So the first step is to cool it down and to bring it to a pre-treatment. And then here you're going to separate the cells from the broth. From there you have 2 possibilities based on the enzyme. I'm going to get into the details, but 2 workflows, as they call it, based on the property, where you either crystallize the enzyme, so production scale crystallization of enzyme, or if the enzyme doesn't work this way, you're going to do ultrafiltration of the enzyme. So first step, removing the biomass. So by biomass, I mean the host from the broth. So you can use flocculants to favor this type of separation. And yeah, better for bacterial fermentation because they are smaller than filamentous fungi. So you add your flocculant here, and it goes to the second step, which is a drum filtration. So a drum filtration, it's kind of, it looks like that. It's pretty big. And what you do is that you have a continuous flow. On one hand, you get the broth, the cells. Already some of the cells have been removed with the flocculation, and you get the protein. And here you get your drum. So what happens, you get here, you put your cells and protein, you have a central duct which does a suction, and then it kind of stick the things to the rolling drum. And then, oops, sorry. And so your liquid, which contains your enzyme, is gonna get in the middle there, and your biomass stay on the outside, and you have this cake, and that's how it looks. So you have tons of cells going through this. So now, like I said, you have 2 workflows for the protein purification based on the enzyme properties. So some enzyme can crystallize fairly easily. So it's the case of proteases, for example. And some do not, and so you're gonna do an ultrafiltration step. So you have filter plate filters, as we call it, and it reduces the salt, gets rid of the small peptide, as well as some other small molecules, because your host is still going to produce a little bit of organic acid, which you need to get rid of. And you remove the water, so you do at the same time a purification and concentration step. And the membrane life is a key cost parameter, because these are really expensive. So how it works here right there at the beginning, you get your broth. You still have some cells, even if you remove them. It's not super pure, and you still have a lot of contaminants. You apply some pressure, your proteins and small molecules gonna go on that side, while your biomass stay on that side, so what you don't want. And here you get your enzyme. So now you have a concentrated liquid formulation. So if we look at crystals, so enzyme crystallize. Obviously, that's how you do 3D structure. It's not possible for all enzymes, and it requires a high purity starting material, which means that at the end of this two-step of cell removal and flocculation of biomass and cell removal, you are still 90% of the protein are the one you want, so you have really high level. And you need the right conditions, obviously, for whoever has worked on trying to crystallize protein for 3D modeling. You know how hard it is, not modeling, but determination. So temperature, pH, and cell concentration. And some enzyme tends to spontaneously crystallize in the broth, and that can be a main issue, because then you're gonna clog all your pipe and your factory is offline for a few weeks, which is bad. You don't want your factory to be stopped. So these are two types of crystallization you can get. So in the broth and when it's concentrated. And you can actually see them on a microscope. So you take the broth and you'll see this crystal of enzyme in the microscope. The final step is granulation. So why do you do granulation? Enzymes are proteins. Proteins can lead to allergies, right? And for the safety of the workers, you don't want all this enzyme powder being everywhere. Otherwise, people are going to develop allergies, which can lead to death. So you have a simple process. So you start with your, not simple, but you have a process with your raw material, enzyme crystals or liquid concentrate. And then you're going to add all the material you need for that. You mix it. Here you kind of sort it a little bit. You dry it, you sort it, you mix other additive, which can be specific to your application. Then you have this mixing coater, like we call it, so where you just mix things together. Then you get on a cooler, because it's usually at high temperature. Again, you sort out things by size, make sure that everything is homogeneous. You don't want big clump and smaller ones. And then eventually you end up with your final product. So you can have powder or liquid, depending on, well, actually on that one we're granulated. So you can have it then into big bags or into little container like that. So the granulate structure, it's simple. You get a core, which is composed of the enzyme, some sodium sulfate, some binder, so just to make sure your enzyme is still inside. I don't remember exactly what Caroline is doing. Oh, sorry. And you have the coating, which is titanium dioxide, kaolin again, and then some PEG. And that's how it looks like. And that's most likely sometimes what you find in your product. This little granule, this little powder can be broken down, but the white powder you'll find in your detergent can be some of the enzymes. Sometimes they add colors. So you know you have a new enzyme in a product when they came up with a new design and you have a little bit in the middle or something, you have a red, white, and blue things usually, or you have a little liquid form as well in some of them, liquid bubble in it. That's usually a new enzyme and a new preparation and a new way to make it. So the granulation tower, it's where you end at the end. So all this process happens right there, and you can see that a big piece of equipment. These things are huge, and when you get to a plant, it's pretty impressive to see this constant flux of enzyme just coming out non-stop. You have all your fermenter, and you see kilograms and kilograms and kilograms of enzyme. And it's really satisfying when you've been in the lab making these strains to go to the plant and actually see this little milligram of enzyme you produced in your test tube being put in a major fermenter. And this one is not a loading truck, but this one, for example, and you see it leave the site in a big truck like that. And then in some cases, you can actually buy it at a supermarket. So you buy what you help making, which is actually pretty cool. Even if it's just for soap, it's still something. One of the main optimization which has been done in the enzyme business have been around the protease. So if you remember back in the day, all your laundry or even your dishwasher were operating at fairly high temperature, right? I don't remember, it was 40, 50 degrees Celsius. And about 10 to 15, maybe 20 years ago, they started this new trend, which was a cold washing, where you don't need to use hot temperature. And this came from the fact that some proteases were engineered to work at low temperature. So it was mainly the progress made on the engineering of the protease to be able to operate at temperature between 10 and 30 degrees Celsius instead of the 50 degrees Celsius which were used before. And this allowed to decrease fairly considerably the energetic demand of a household. So through little improvement like that, you can also not only get good product, but also help the environment by decreasing the energy demand. And also reducing the amount of chemicals we can put. So as a conclusion, industrial constraint will market intellectual properties. It's a battlefield out there when it comes to enzyme. There's a lot of competitors, and whatever you do, always start, if you want to do a process, by that. Because it's really painful to have spent years and years of development and get to a product, and suddenly you cannot sell it because a competitor is going to sue you. And that happens. And people get really, really creative when it comes to looking at intellectual property and make sure you did not infringe a patent. For example, the sequencing now is down. The technology is so good that you can take any of this product, extract the DNA, and obviously by proteomics you will know what is the enzyme, what the sequence, but you can also know which host was used for that. And there are some cases where it was found out that some of the strains used by competitors were coming from patented strains, or in that case of Novozymes. So Novozymes are the strains which was patented, a competitor used it, produced the enzyme, put it in an application, so in that case it was dish soap, and we were able to go look at the DNA evidence found in this dish soap and be able to reconstruct the genome and found some key mutation done on the host which were patented by Novozymes. So that's a little bit scary, but that's what people are willing to do to make sure you are not infringing their patent and not taking their intellectual property. So obviously you have cost, and cost. I mentioned cost, process and safety. And then the physical constraints, which are stability through the process and the application, and the ease to clone and express. So finally, some scientific things right there, but mainly it's bioprocess and big money. So if one of these constraints is overlooked, it could be a waste of resources, because if, for example, again, if it's not safe and workers at the factory start dying, that's not good. We all agree on that. Or if it kills you when you use your dish soap, that's not good. So it's always good to make all this. And even if we are not pharmaceutical, you still have toxicology studies. So you take the enzyme, feed it to a rat, and make sure they don't die. So that's something to keep in mind because of the regulatory constraints in the different markets. And I think I'm done with that. So here's my email address. If you have any question, don't hesitate.